Good morning. Good morning, one and all.
Good morning, sir.
Good morning, sir.
Can we start?
Yes, sir.
Kumar, sir, can we start? बापू साहेब आले का? हो हो. बापू साहेब
नाही म्हटलं 5 मिनिटं मी थोडं 5 मिनिटं.
सर करायचं का चालू बापू साहेब?
हो चालू करा ना सर. 100 पार्टिसिपेंट झाले. सुरू करा. सुरू करा सर.
ओके ओके. Good morning, one and all. For today's national webinar on Archegoniate and Mycology, organized by Department of Botany, MSG College, Malegaon-Camp. As we know, we all are going through the universal crisis, that is Corona pandemic. During this period of students, to overcome this loss, we are always trying to engage the student by virtually, and the eminent personalities today we have invited for the overcome this loss. The today's eminent personality, Dr. Adarsh Kumar, retired principal, Feroze Gandhi College, Raebareli, and the second eminent personality is Dr. M.N. Bhajbhuje, principal in charge of and head of the Department of Botany, Jawaharlal Nehru College, Nagpur. For today's webinar, our respected chairperson, Principal Dr. D.F. Shirude sir, our Vice Principal Dr. C.M. Nikam, Dr. N.B. Bachhav, our Head of the Department, Dr. J.T. Jadhav sir, Dr. Vaishi Shastri, Dr. Mamude sir, and all my colleagues and the respected dignitaries who joined the webinars, and my dear students. Today, we are going to conduct the webinar on Archegoniate and Mycology. For the detailed information about the webinar, I would like to request Dr. J.T. Jadhav, Head of the Department, to say some word about this webinar. Dr. J.T. Jadhav.
Hello, good morning to all. Respected Principal and Chairperson of today's national webinar, Dr. Dinesh Shirude sir, Vice Principal, Dr. C.M. Nikam sir, Academic Coordinator, Dr. N.B. Bachhav, today's resource person, Dr. Adarsh Kumar and Dr. M.N. Bhajbhuje, IQAC Coordinator, Professor S.I. Ansari, NAC Coordinator, Dr. Mukund Bandari, all the staff members and my friend participants. With great joy and immense exultation, I feel privileged to extend my warm welcome to all presented here for this virtual national seminar. Well, today we are all gathered here for a national webinar on Archegoniate and Mycology, which is organized by my PG Department of Botany. Our department has tradition to arrange conferences, workshops, and webinars. This year, we have arranged a state-level webinar on fishery management on 4th October 2021. Today, we are organizing this webinar to fulfill the need of UG and PG students. Once again, I welcome all the dignitaries and participants and the resource persons. Thank you. Over to Dr. P.S. Patel.
Thank you, sir. Thank you very much. Now, I request to our Honorable Principal, Dr. D.F. Shirude, sir, for Principal's address. Dr. Bapu Sahib Shirude.
May I audible, sir? May I audible?
Yes, sir. Yes, sir.
Good morning.
Good morning.
Respected Dr. Adarsh Kumar, respected Dr. Bhajbhuje, sir, Vice Principal of our college, Dr. C.M. Nikam, sir, Academic Coordinator, Dr. N.B. Bachhav, sir, and Head of the Department, Dr. J.T. Jadhav, sir, all participants and students, on the behalf of Mahatma Gandhi Vidyamandir, I welcome all of you. First, Happy New Year to you all. It is always a thing of happiness to see that the Department of Botany arrange activities for the students and lecturer and teachers, particularly when the elite guests are called upon to deliver lecture and enlighten the student for their better future. I am sure that the national webinar will be taken sincerely by the student and participants, and they would surely be benefited by this feast and knowledge which would make them ultimately wise. The guests who have accepted our invitation and sparing their valuable time for our students, I, on behalf of our management and college, express my gratitude and hope the cooperation in future also. I appeal to all participants and dear students to participate actively in this national webinar organized by Department of Botany. Thank you, sir.
Thank you very much, sir. Your inspiring words are mean lot for us. Thank you very much. Now, I request to Dr. Y.C. Shastri to introduce today's resource person, Dr. Ashok Kumar.
Good morning to one and all. Respected Principal Dr. D.F. Shirude, sir. Respected Vice Principal Dr. C.M. Nikam, sir. Academic Coordinator Dr. N.B. Bachhav, sir. IQAC Coordinator Professor S.I. Ansari, sir. NAC Coordinator Dr. Mukund Bhandari, sir. Head of the Department of the Botany Dr. J.T. Jadhav, sir. All the staff members and participants. Now, let me introduce today's resource person, Dr. Adarsh Kumar. His qualification is MSc PhD in Botany. He completed his research work, PhD work, under the guidance of eminent bryologist Dr. Ram Udar. He is associate professor and at present he is associate professor and retired principal of Feroze Gandhi College, Raebareli, Uttar Pradesh. Dr. Adarsh Kumar is a member of Board of Studies and ex-member of Academic Council of Lucknow University. He is a life member of the following bodies: life member of Indian Journal of Environmental Protection, life member of Indian Society of Remote Sensing, Dehradun, life member of Indian Biological Society, University of Lucknow, life member of Birbal Sahani Institute of Paleobotany, Lucknow, and member of Academy of Environmental Biology, Lucknow. Paleobotanical Society of India, Indian Botanical Society, and Indian Society of Remote Sensing conferred fellowship to Dr. Adarsh Kumar. Dr. Adarsh Kumar is a research guide, and two students have completed their PhD under his guidance. Two research projects to his credit. He is the author of two books, namely Bryophyta for degree students and Bryophytes in relation to the air pollution. Dr. Adarsh Kumar published 51 research papers in reputed national and international journals. Dr. Adarsh Kumar presented papers in 32 conferences. This is his introduction in nutshell. Thank you.
Thank you, sir. Now, I request Professor Atul Wagh to introduce today's next resource person, Dr. Bhajbhuje, sir. Atul Wagh.
Thank you, sir. Thank you for the opportunity. I would like to thank and introduce Dr. M.N. Bhajbhuje, sir, currently in charge principal and professor and head Department of Botany, Jawaharlal Nehru Arts, Commerce and Science College, Wadi, Nagpur. Sir completed his MSc in 1986 from RTM Nagpur University, and after all, in 1989, sir completed his PhD. His research area is seed pathology, fungal biotechnology, aeromycology. Sir has huge experience of 32 years at UG and PG level. Sir has administrative experience as well as sir is invited as a faculty. Sir is academic counselor in Indira Gandhi National Open University, Nagpur Center. Various research projects are undertaken by sir. Various books as well as research papers were published. Currently, sir had completed his 328 online conferences only in the lockdown period and 71 offline conferences seminars. Sir had completed his activities and various medal receipts. First, the thing is recipient of Best Teacher Award by VSPM Nagpur. Received certification of appreciation for successful organization of UGC sponsored All India Seminar. Dr. Jagdish Chandra Bose National Award for Excellence in Botany, and a fellow member of Research Innovations. So I am very happy with this eminent personality. Please welcome Dr. Bhajbhuje, sir. Thank you very much.
Thank you, sir. Now, I like to request Dr. Ashok Kumar for his Adarsh Kumar for his presentation. Dr. Ashok Kumar, sir. Adarsh Kumar, sir. याला फोन लागो सर दादाला फोन लावा सर दादा नंबर आहे दादा सर
Very good morning, everybody. Dear Dr. D.F. Shirude, Chairperson and Principal of the college, Dr. C.M. Nikam, Vice Principal, Dr. N.B. Bachhav, Academic Supervisor, Dr. J.B. Jadhav, Head Department of Botany, Dr. Y.C. Shastri, Coordinator of this program, all the members of this organizing team, Dr. M.N. Bhajbhuje, the learned speaker on mycology, my all dear students and friends who are attending the webinar. Today my talk is on the Archegoniate. My dear friend, Archegoniate includes those plants which possess archegonium, female reproductive organ, that is egg, and its accessory cells enclosed within the sterile jacket. My talk is entirely based on as per the syllabus of the Pune University as sent by your faculty members. The Archegoniate, especially the cryptogams, includes Bryophytes, Sensu stricto, and Chloromycetin Sensu stricto as per by the Campbell 1905. My dear friends, regarding the gymnosperms, they are also a part of Archegoniate Sensu lateral. Regarding the angiosperms, in primitive angiosperms, the archegonium is highly reduced form. All these groups are or have the archegonium as a female reproductive organs. On the other hand side, those plants which are simple, primitive, and without the archegonium, they are called aarchegoniate. Aarchegoniate is where archegonium is absent includes the Thallophytes, that is the algae, fungi, lichen, etc. Next slide, please. After the archegoniate, the other part which is very important for the students, that is the embryophyta. Those plants which bear the embryo fall under the embryophyta. It includes bryophyta, pteridophyta, gymnosperm, and angiosperms. My question is for the students that what is the embryo? If you see in this picture, this zygote, which is formed by the fusion of the male and female gametes, it forms the zygote, which is the diploid in nature. If this zygote divides by the mitosis, it forms a mass of diploid cells, which is called the embryo. This embryo then differentiates. In case of the bryophytes, the sporophyte consists of the foot, seta, and capsule. And in the pteridophytes, the sporophyte has true roots, true stem, and the true leaves. While those plants which come under the thallophytes are archegoniate, and these plants are the without embryo. My next slide, please. Here, bryophytes. My dear students, when you are very young and child stage, your parents told you A for apple. They show you the apple and then you learn the A. Similarly, but for bryophytes, B, you should remember the plant funaria. It's very unique plant. To understand the bryophyte, always remember this plant funaria. This part is the gametophyte, which is the dominant in life cycle. It has three parts. The rhizoids, which are multicellular, they absorb the water and mineral nutrients, then axis, and on the axis, the spirally arranged the leaves are the present. At the apex, it bears either the arteria, that is the male sex organ, the collectively known as the male head, or it contains the archegonia, and that part is known as the female head. So this part is the female head in which the archegonia are the present. After the fertilization of the archegonia, the zygote is formed. This zygote divides by the mitosis and forms the embryo. My dear friend, bryophytes are unique in nature. In these plants, the formation of archegonium starts for the first time. In these plants, the formation of the embryo starts for the first time. This embryo differentiates and form the foot, which is embedded at the apex of the female shoot, then long seta, and the apex of long seta, there is the capsule.
Yeah,
So
excuse me.
this capsule,
Actually,
this
slideshow. Sorry to interrupt, but slideshow is not changing, or maybe slides can actually be. Slideshow is not started yet, so the slide is not changing.
I'm just showing the. Okay, is it visible now?
It is visible. Can you also start the slideshow
Yeah.
from the slideshow menu?
Can you see it?
I mean, if you start the slideshow, then it becomes full screen. It will be more visible.
Can you see it now?
We can see it, but it is the slide number 1 which is on the screen.
Okay, can you see amplifier?
Yeah.
Yeah.
Yes.
So in bryophytes, the male plant body is the gametophytic, and the sporophytic pair, which consists of the foot, seta, and capsule, depends on the gametophyte for the nourishment and for the support. This is the unique feature of the bryophytes. If you recall the bryophytes, always remember the plant funaria. The main plant body is gametophyte, and sporophyte depends on this gametophyte. Next slide, please. Here I am showing the classification of each. This classification falls under the category of the phylogenetic system of the classification. Ischler divided the plant kingdom in two subkingdoms, the cryptogams and the phylogams. Cryptogams includes those plants where flower is absent, the seed is absent. This cryptogams divided in three divisions: thallophytes, bryophytes, and pteridophytes. And on the other hand side, phylogams divided in two divisions: gymnosperm and the angiosperms. This placement of the 5 divisions is as per the phylogen. It means the most primitive group is the thallophytes, from which evolved the bryophytes and then pteridophytes. My talk is based on the Archegoniate, that is on bryophytes and the pteridophytes. If you consider the bryophytes, these bryophytes are more evolved than the thallophytes. And other hand side, the pteridophytes are more evolved than the bryophytes. Not only this, the thallophytes and bryophytes, both divisions show affinity in their gametophytic stage. On the other hand side, bryophytes and pteridophytes show affinity mostly in the sporophytic stage. Next slide, please. This is the Smith classification, which is prescribed in Pune University. Here, division Bryophyta has been divided in 3 main classes. One is the Hepatica, other one is the Anthocerote, and third one is the Massai. As per the ICBN, International Code of Botanical Nomenclature, the new name of the Hepatica is the Hepaticopsida. The ending of class is Opsida. Then Anthocerote is known as Anthocerotopsida. And similarly, Massai is known as the Bryopsida. My dear friends, the recent classification proposed for the bryophytes is somewhat different. As per this classification, Hepatica has been placed in separate division, and that is called the Markensiophyta, as per the Stotler et al. 208. Similarly, the class Anthocerotopsida has been placed in another separate division that is called the Anthocerotophyta. This is by Stotler and Stotler 2005. And on the other hand side, Massai, Bryopsida, which is known as the Bryophyta. It includes all the mosses. This division has been proposed by the book and the Goffnet in 2000. The Hepatica includes the following order: Spirocarpales, Markensiales, Jungamaniales, and Calopriales. On the other hand side, the Anthocerote or Anthocerotopsida has a single order, that is the Anthocerotales. While the mosses, Massai, Bryopsida or Bryophyta, this is the largest group. So there is a concept of the subclasses. And these subclasses are the Sphaeridae, Andreidae, and Bryidae. The Bryidae has 2 orders, Polytrichales and Bryals. Andreidae has Andreals, and Sphaeridae has Sphagnales. So this is a brief concept of the Smith classification, which is in your syllabus. Now I'm talking about the origin of the bryophytes. This is still controversial. It is an abominable mystery. This term, abominable mystery, was termed by the Charles Darwin for the angiosperm. Similar case lies in case of the bryophytes. There is no such perfect evidence on the basis of which we can say that these plants are the original plants from which the bryophytes evolved. In spite of that, there is no fossil record to establish the origin of bryophytes till date. There are two views regarding the origin of bryophytes. One is the Algal Hypothesis proposed by Lindner 1903, Bauer 1908, and Fritsch 1960 and 1945. The other thought of the school believed that bryophytes evolved from the pteridophytes by descending. So this is called the pteridophytean hypothesis. The supporter of this theory are the Scott 1911, Kashyap 1919, and Haskell 1949, and Mehra 1967. Here I am giving some example of the algal hypothesis. This is the coleochaete, green algae. The plant is thyroid in form of the rosette, gametophyte. After the fertilization, the zygote is formed in this plant. In case of algae, this zygote soon divides by meiosis, reduction division, and forms the spores. It is believed that in due course of evolution, this zygote of algae fails to divide by the meiosis or reduction division, and it starts to divide by the mitosis. And when zygote divides by the mitosis, in last slide I have shown it forms the embryo. And this embryo differentiate and form the capsule, and its example is the rixia. In case of rixia, this is the bryophyte in which sporophyte has neither foot nor seta, only sporogonium or capsule is present, which is found embedded within the thallus, especially in mid-dorsal groove, as in case of in this coleochaete where the zygote is embedded within the cell. So it is believed that the bryophytes evolved from simple algae like the coleochaete. The other example is of the cara. This is the sex organ of the carga. This is the female sex organ, nucleus, and at the base there is the male sex organ, the globule. These two terms were coined by the Smith. If you observe in this male sex organ, this is the antheridium filaments, and at the upper side there is a covering of the shield cells. This antheridium. This antheridium is converted in the antheridium of the bryophytes. But the origin of this structure and the origin of this structure in bryophyte both are quite different. So Smith coined a new term, globule. He believes that from the globule the antheridium arises. Similarly, in case of the nucleus, which is the female sex organ of the carga, the egg is present in this center and it is invested or covered by the shield cells. At the apex, there is the crown. This covered or protected structure of egg is similar in this case. In the Archegonium, the outer is the sterile covering. In the base, there is egg, and these are the accessory cells, ventral canal cells and neck canal cells. So it is believed that from the nuclear structure, the Archegonium evolved. That's right. This is the angular theory. Supporter of this theory believe that the sex organs of bryophytes, where the sterile covering is present, evolved from the brown algae Ferophytes. My dear students, in Ferophytes, there is 3 subclasses. One subclass is isogenerating. Its example is Ectocarpus. The other is heterogeneity, and third one is the cyclospore. So I am giving the example of Ectocarpus, which is the member of subclass isogenerating. The supporters of this view believe that the Anthidea and Archegonia evolved from the plurilocular sporangium. This is the plurilocular sporangium. In this sporangium, all the cells are fertile. They produces the mitozoespores. In due course of evolution, the outermost part becomes the sterile and it forms the covering. As you know, that in case of the development of male sex organ, that is the anthidium, it is more active and number of cell division increases. On contrary to this, the development of the female sex organ, which is passive, the number of cell division decreases. So in due course of time, the outer layer become the sterile and they do not form the mitozoespores, and the number of division increases, and all these cells are the fertile endogonial cells that form the anthozoites or spores. The other plurilocular sporangium, the outer covering form the sterile covering, and the inner side, the cells divide in fewer number. It's more passive. And the last cell become the egg. This is the ventral canal cell, and these are the neck canal cell, and thus the archegonium is formed. So it is believed that the sex organ of Anthidium and Archegonium evolved from the brown algae, that is from the isogenerative, that is from the ectocarpus. This is the second theory. This theory is pteridophytean hypothesis. The supporter of this theory believe that bryophytes are descendants of the pteridophyte. The reduction division took place in pteridophytes, and by the reduction, the bryophytes evolved. In support of this view, this plant is of the Hardiphyton. This is the gametophyte, and on this is the sporophyte, where the dichotomy is visible, and these are the sporangia, where the spores are produced. In due course of evolution, this dichotomy stops, and the apical meristem comes downward at the middle part, which becomes the meristematic in case of the anthoceros, and the lower part is the foot. So from the sporophyte of the pteridophyte, the hornworts as well as the mosses, the sporophyte evolved. The next slide, here the stomata on the sporophyte of the Hardiphyton are visible, and these are the stomata on the anthoceros are also visible. That the sporophyte bears the stomata, and it possesses the photosynthetic pigments and form the starch as its reserve food. Other than above two theories regarding the origin of bryophytes, there is a third view of Church 1990, which is neither tenable and nor acceptable due to certain well-established drawbacks discussed and proposed by the French in 1931. Actually, Church in 1919 believed that the earlier, when whole earth was covered by the sea, marine water, there was no land visible, and the plant evolved in sea, that is the sea plankton, and for its coined a term thalassiophyta. These are those plants or phytoplanktons which evolved in the ocean. From these thalassiophyta, some land appeared on ocean, and these plants become the benthic. So this is the sub-aerial, the transmigration plants are benthic plants. and from these plants the land flora like bryophytes evolved. But this theory has not been accepted by anybody. Till date, there are two views regarding the origin of angiosperm. One is the algal hypothesis and other one is the pteridophytean hypothesis. The second part of my talk is the contribution of Professor Ramudar. Professor Ramudar was the student of Professor S.K. Pandey. And Professor S.K. Pandey was the student of Professor S.R. Kashyap, the father of Indian biology. Actually, S.K. Pandey was the student of Professor S.R. Kashyap, and Professor Ramudar FNA was the student of the Professor S.K. Pandey. Professor Ramudar, born in district Basti of Uttar Pradesh, after completing his intermediate, he joined University of Lucknow and did his BSc, MSc with honors, and joined his PhD work under the eminent biologist Dr. Professor S.K. Pandey. After completing the PhD, he joined as a lecturer in University of Lucknow, and from that time, he established the well-established biology lab at University of Lucknow. His main contribution is in taxonomical studies. He studies the members of Bacteriales, family Jungamaniaceae, Furinaceae, Legionnaceae, Notothylaceae, Cythodium, Candonanthus, Notoscyphus, and so many plants. He reported several genera from India for the first time. These plants are Calobrium, Buxbaumia, Conicolia, Cerendocolia. He reported so many new species. I have mentioned some: Rixia pandei, Rixia reticulata, Plazocosma, Poriana, Mania, Porenai, Mania, Personae, Cythodium denticulatum, Ricardia, Santapoi, Buxbaumia himalayensis, and Calobrium indicum. He completed several morphological studies, such as in family Sortariaceae. He completed the monograph of the Athlemia pinguis and Plazocosma intermedium. Not only this, he completed the palynological work of the liverworts and the hornworts, which are very useful to solve the taxonomical problems of the bryophytes. He started his work with the cultural studies, and he did the cultural studies in plant Rixia. He also completed some work on the physiological studies and reported several enzymes in case of the hepatic. He studied the oil bodies, which are found only in the liverworts. And these oil bodies are present only in the living samples. He published two famous books. One book is An Introduction to Bryophyta for the students, and other very, very famous book, Biology in India, which is useful for the PhD as well as for the research purpose. Under Professor Ramudar, following students completed their PhD on different topics. His first student was the Dr. Vinod Chandra Bhatnagar. He retired from the University of Delhi. He completed his research work on Raebareli. Then, second student, Dr. S.C. Srivastava, who retired as a professor and head of the University of Lucknow, and he followed all the trends laid down by the Professor Udar, and he continued the same lab. His main work is on the Mediterranean and the flora of different part of the India. The third student, Professor Dr. Dinesh Kumar, his work is on the Calobrias. Professor Dinesh Kumar also did some work under Professor P.N. Mehra from the Chandigarh University. Then Dr. Virendranath, his work is on Furinaceae. He retired from the National Botanical Research Institute, NBRI, Lucknow, as a deputy director. Then Fifth one is Dr. D.K. Singh. His topic was the notothylaceae. He retired from the Botanical Survey of India, Calcutta, as a director of BSI. Then Dr. Asha Gupta. He retired from the Birbal Sahni Institute of Paleobotany. His specialization was on the palynology of the bryophytes and especially the liverworts and the heartworts. Then Dr. U.S. Avasthi. He was in Associate College of the Lucknow University and his topic was the Trichanthoidae. Then myself, Dr. Adarsh Kumar, my thesis topic was the Indian jungle history. Then Dr. Farhad Sahin, his topic was Porelaceae. He is serving in the forensic lab. Dr. Dhirendra Kumar, he was in Kanpur University and he also did some work at Allahabad University under Professor D.T. Pant. His topic was the Redulaceae. Then Dr. Geeta Asthana. Geeta Asthana retired in 2019 from University of Lucknow as a professor and head of the Botany department. His PhD topic was the Chloromycetin. Then Anita Agrawal. At present, she is in the USA and her topic was the Legionnaceae. And at the last, the student of Professor Udar is Dr. Ashish Asthana. He is still working in National Botanical Research Institute, Lucknow, as a senior scientist, and his topic was the Anthoceros. So thank you very much. This was my whole talk on the Archegoniate and the contribution of the Professor Ramudar. Okay, thank you.
Thank you, sir. Thank you for your valuable, informative, and quality guidance. Your contribution in the field is remarkable. Thank you, sir. Thank you very much. Now, I would like to request our second eminent personality, Dr. Bhajbhuje, sir, for the presentation. Bhajbhuje, sir.
Can you hear me?
Yes, sir. Yes, sir.
The topic which is chosen for today's seminar is the beneficial aspect of the fungi. Generally, we think that the fungi are pathogenic, but so many aspects are there which give the benefit to the human beings. This is the preliminary part. I will just directly come to. These fungi are classified on the basis of their mode of nutrition into parasites, means they survive on the living. Either they be obligate or maybe the facultative, total or the partial. They may be hyperparasite, means parasite may again survive on the body of the parasite. The third category is very useful. They are saprophytes. It's a group of fungi survive on the dead and decaying matter. This is the very important part. If there is dead and decaying matter, this should be decomposed, and only decomposers are the saprophytes. If there would have been no saprophytes, then the material which we consume and lost and remains, they will remain as such without their conversion into complex to the simple forms. Harmful to the simple ones, harmless one. And again, one category is there, symbiont. Some of the fungi are symbiont. They form the association ligand. Some of the fungi, they also remain associated with the roots of the higher plants and also provide the nutrition and the food to the higher plants in their deficiency. This is our problem. Fungi cause the diseases. These are some of the photographs showing all the material of the plant is infected. White red disease caused by the albigo candida, which reduce the which reduce the productivity. Potato. Potato is a product. Now it has more demand than the any other product consumed in the market. Potato chips. Everywhere in the we consume the material, the potato is one of the important ingredient. Third, wheat. This disease is caused by Vaccinia. All indicate these diseases reduce the productivity, they lower the production. If these are taken out, the late blight of potato is called by Phytophthora. If is taken out, Phytophthora is taken out, Vaccinia is taken out. And if they are used for production of the metabolites, they will be more useful. We know that these are the harmful activities of the fungi, dairy product. They spoil the product. They spoil the mucor, penicillium, fusarium. They spoil the dairy product like milk, like cream, like butter. Bread spoiled by the mucor, aspergillus, neurospora. These are the photographs showing they are the food spoiler. Hence, we cannot reject them. Because these fungi, Penicillium, mucor, aspergillus, neurospora, they are also useful in other way. How they are useful, we will discuss later. Now, this is the photograph. Spoilage of fruits. First photograph is of apple. It is totally spoiled by a penicillium. The name is very important, penicillium. It is a penicillium chrysogen. This food, it is a apple spoiler. But how this penicillium is important in your life? Penicillium given a job, see the sentence is, penicillium given job to the millions of peoples. They secured the life of millions of people. How the sentence is? The one fungus which caused the disease, but the same fungus secured the life of the millions of people. How it is possible? I will describe it. This is the strawberry. And this is happening only because of our negligence. Because we are not taking care. We are trying to allow them to grow in that medium. See this banana. Very useful fruit. What is happening here? Generally, we have habit to bath under shower and we enjoy it. If moisture remain on the skin, then aspergillus may cause aspergillosis in the skin. Hence, it is necessary to clean the whole body with any other components. there is a chances of the ringworm. These are the 3 photographs I have shown you. This is the 3D picture from the net. Ringworm caused by Mycosporium. Blastomyces caused by blastomycosis caused by blastomyces. There are the ear problems in youngsters. This is also caused by the Aspergillus nodulans. Means one has to take care. These microbes should not allow to grow. One should not provide them to grow. Provide them, do not provide them a climate. This is the slide. What are the benefit aspect of the fungi? Forget about the diseases. Number 1, fermentation technology. Alcohol production. It is a need of ours because medicine faculty, we call medical faculty, need alcohol at every moment for the process of sterilization. Now there is one word which is used, sanitization. This word came into existence in the corona. It is a sterilization word. And for sterilization, alcohol is a must, or room sterilization, or laboratory sterilization, or hand sterilization. And fungi play important role in fermentation technology, production of the alcohol from waste material, which is not useful material to the human beings. This is waste to the human beings. How it is useful? Beverages. There are 2 types of beverages. Alcoholic beverages. Very sorry to say, alcoholic beverages have great demand at international level than any other product. And non-alcoholic beverages, there are 2 types, tea and coffee. This tea and coffee is also has a great demand because in morning hours, we are habitual of either tea or coffee. Organic acids. In our body, several reactions going on continuously. And during aerobic respiration, glycolysis takes place. It follows the Krebs cycle. And then there is electron transport system, ETS. During the Krebs cycle, so many acids are produced, hence it is called the organic acid cycle. And first acid which is produced is the citric acid, which is also called the acidulant. But suppose the body is not properly working, then there should be a supply of organic acid from outside source. Then which is the source? Who will produce same type of the organic acid? Other body. The other body, why the other body will give a same organic acid to the other person? It will not. Enzymes, vitamins, and for this, the fungi is the best source. Fungi produce organic acid, several types of organic acids. Fungi produce several types of enzymes, vitamins. You know, the best example is the insulin. Insulin can be constructed. biotechnology method. Then next one is a fermented food. Confectionery and bakeries. Number of hospitals, if you count the number of hospitals in our Maharashtra, then in India, you will find the patients are provided with a bread in the morning. How much quantity of bread is required every day? It is a fermented part. And if the fungi involved in fermentation, then it will become a beneficial aspect. Food processing. We need a food which is easily digestible nowadays, particularly for the senior citizens. What type of food they need? They need idli, they need dosa. It is a South Indian products, and maximum South Indian products, they are the fermented products, very famous product. Beside this, so many fungi. I was talking about the penicillium. It produces antibiotic. It produces antibiotic penicillin. And discovery of penicillin given the job to the unemployed youth. Several unemployed across the world after discovery of the penicillin in 1929, and when all the tests were completed in 1940. Then in 1941, the industries were started. And it saved the lives of the patient, those who were affected with the bacterial disease. This is the penicillin. After that, more than 4,000 antibiotics have been discovered after the discovery of penicillin, not before discovery of penicillin. Then disposal of waste. This is the photograph, disposal of waste. We generally consume the product and dispose it. जितना हम खाते हैं, जेवढं आम्ही जेवण करतो तेवढंच बाहेर फेकतो. वेस्ट जातं, खाल्ल्याच जात नाही. तुम्ही पहा एखाद्या जर तुम्ही लग्न समारंभ किंवा कुठे तुम्ही जर गेले तर तुमच्या लक्षात येईल की जेवढ्या लोक जेवण करतात तेवढं वेस्ट जातं ते फेकल्या जातात म्हणजे कुठेतरी टाकल्या जातात. हे असं who will convert this harmful into the harmless one? The fungi involved, particularly the saprophytic fungi involved, and make them into harmless products like a manure. Now, see, this is a man. This is a photograph of Alexander Fleming. I was talking about the penicillin. This is basically a medical doctor. And he worked as a doctor in battlefield. And he noticed that most of the soldiers, they died due to small injury caused by bacteria, particularly streptococcus, streptococcus. And he was thinking about some medicine, and he was working in that direction. But what happened? One day, This is the laboratory. He worked in this laboratory. This is the laboratory. This is Mary's hospital. I will talk about later. Then accidentally, in 1929, I'm talking about 1929, he was working on the bacteria Streptococcus. He has inoculated the bacteria on the petri dish. Bacteria grew at 45 degrees Celsius and forgotten to incubate them or incubate the petri dishes in incubator, thermostatic incubator. And he had gone for vacation, 2-week vacation. After returning to the laboratory after a week of time, he noticed that he has forgotten to keep this inoculated material to the oven for their growth. And he was very puzzled that my experiment is lost. When he visited the laboratory again, then he find some filaments were growing on the petri dishes instead of the bacteria. He has taken out these filaments and grown these filaments in a broth medium and then trying to identify what is the filament is. Then finally he concluded that it is a filament of penicillium. It is a fungus. Actually he was working on bacteria, not on the fungus. It is a chance. And then he allowed to grow this filament in a broth and culture filtrate solution of the broth. It became yellowish within a week of time. 1 or 2 drops of the broth added on the culture of bacteria. Then bacteria killed. Then he realized that the material which is secreted by the penicillium may be toxic to the bacteria. This is the concept developed. Then he has taken the help of Fleury and Chen. Both were biochemists. They had, I think somebody is not interested. If you are not interested, then I will stop it.
Any problem, sir?
Who is trying to disturb the screen?
Okay.
Somebody were trying to disturb the screen.
सर स्क्रीन पुन्हा तुम्ही पुन्हा शेअर करा स्क्रीन.
नाही म्हणजे आता हेच काम करत राहायचे का?
नाही कोणीतरी
मग त्याला कंट्रोल करा ना तुम्ही.
हो रिमूव्ह करा.
रिमूव्ह करा त्याला, हाकला त्याला. पनिशमेंट घेत काय झालं? अरे आम्ही काय सांगतो, काय इथे काय पागल बसलो का इथे?
Please continue, sir.
And then there was a discovery of a penicillin. This is the photograph. This man, important is, there is a demand of antibiotic because this penicillin was effective on the bacteria, and there was a demand of this antibiotic across the world and continues to outstrip supply of Holland till Howard Walter Fleury and Ernest Boris Chan, two biochemists, have given it. And in 1941, the factories has been started. This discovery given the birth to the American pharmaceutical companies in 1940. Today now, more than millions of people, they are working in that company. आज काय करून राहिले? कोविडशील्ड हे आपण पाहतो. And for this discovery, the Alexander Fleming acclaimed along with the 25 honorary degrees, 26 medals, 18 prizes, 13 doctorates, membership in 87 scientific academic journals, and they share the Nobel Prize. 3% share the Nobel Prize. Fleury, Chan, and Fleming. Nobel Prize shared in medicine and physiology in 1945. This person died on annual March 1955 and buried in crypt in Paul's Cathedral in London. Then what is the important part is, penicillin was discovered by Alexander Fleming. This was the concept. But the article published by Kanwaldeep Singh in 2003, December 2003. And in that article, it is an article of 7 pages. The title of this article is The Story of Penicillin. It was actually John Tyndall. This is the statement quoted. It was actually John Tyndall and not the Fleming who first observed that mold could destroy bacteria. This was in 1875. Means earlier, nearly 55 years earlier, it was discovered, but it was remaining dark. कदाचित येऊ शकलं असतं की त्याच वेळेस ते थोडंसं आणखी पसरलं असतं किंवा माहीत झालं असतं लोकांना. तर त्या 50 वर्षामध्ये जेवढे लोक बॅक्टेरियाने मरण पावले ते सगळे वाचले असते, त्यांचं लाईफ वाचलं असतं. कारण आज पहा तुम्ही, पेनिसिलिन इंजेक्शन दिलं त्याला, पेनिसिलिन खूप डेरिव्हेटिव्ह आहेत, पेनिसिलिनची आणि खूप पेनिसिलिन सुरू आहे तिकडे तिकडे. बॅक्टेरियल डिसीज पटकन क्लिअर करतो. They save the lives. Among these, then there is the discovery of streptomycin by the Streptomyces grassus. Then chloromycetin, this is available in the market. Ergot, The fungus which is called Claviceps purpurea, very readily available in the Himalayan region. It is used as, this fungus is used, this powder is used in contraction of uterus. It is also used in contract of blood capillaries. Ephedrine, which is used in asthma and nasal troubles, is obtained from the yeast cells. When it is synthesized by the yeast cells from the benzaldehyde. Sterols is the organic substance with the hormonal properties. It is secreted by so many fungi, and presently the sterols are used as a medicine. Vitamins is obtained from the several fungi which are now available in the market. There is a production of gallic acid and gluconic acid, which is a need of ours in the body of the patient. It is obtained from Aspergillus, Penicillium. Both are the very common fungi. Aspergillus niger produce citric acid. Generally, the citric acid is obtained from the lime, limbu. किती मिळतं त्याच्यात? Hardly 10%. The species of Aspergillus niger, mutant species of Aspergillus niger, produces 90% citric acid. There is a bioconversion of sugar into citric acid up to 90%. This is experimentally proved. Enzymes, invertase enzymes. It is produced by the yeast cells, Saccharomyces. Zymase enzyme. Zymase enzyme is mostly used in fermentation of sugar into alcohol. Invertase enzyme, they are used for preparation of confectionaries, papers, sugary substances, syrups, medicines. And you will surprise to know these companies from the Denmark and Holland. दोन कंट्रीची नावं या ठिकाणी तुम्ही लक्षात घ्या. डेन्मार्क अँड हे छोटासा कंट्री आहे, डेन्मार्क अँड होलंड. Denmark produces 50% enzymes sold in international market. आज इंटरनॅशनल जगातल्या इंटरनॅशनल मार्केटमध्ये जेवढे एन्झाईम लागतात त्याच्यातले 50% एन्झाईम एकच कंट्री प्रोड्यूस करते, डेन्मार्क. त्या कंट्रीची इकॉनॉमी कशी असेल? होलंड, सेकंड कंट्री, इट प्रोड्युसेस 20%. आता दोघांनीच तर 70% करून टाकलेला. रिमेनिंग बाय द अदर कंट्रीज. What type of enzyme they produce? They produce hydrolytic enzymes, which is used in preparation of soap. ज्या सोप तयार होतात, डिटर्जंट तयार होतात, फूड प्रोसेसिंग मध्ये जे लागतं, जगाची जी रिक्वायरमेंट आहे, द रिक्वायरमेंट ऑफ अक्रॉस द ग्लोब ऑफ द कंझ्युमर्स, it is 50% fulfilled by the single country, Denmark. 20% by Holland, and remaining 30% by the other countries. There is a continuous production. कारण तुम्ही झायमेज एन्झाईम जर एखाद्या शुगरी सबस्टन्स मध्ये टाकले की इमिडिएट अल्कोहोल प्रोडक्शन सुरू होतं. इट इज अ फास्ट प्रोसेस, ओनली एडिशन ऑफ झायमेज. नॅचरल प्रोसेस नाही होत. बट when these substances are available, these products are available from the fungi, these should be taken out, derived from the fungi and used for the fermentation technique. Aspergillus, it produces amylase enzyme. So many products are produced by Aspergillus, and it is used in manufacture of alcohol. Gibberellin, it is a phytohormone. For flowering, gibberellin is necessary. for growth of the plant is necessary, and it is obtained from a one fungus, Fusarium monoliforme. On this basis, the name of this Fusarium monoliforme has been changed to Gibberella. Now these are the products. माहिती आहे तुम्हाला product. Fungi and fruit press, idli, dosa, uttapam, jalebi. आता हे का सांगतो आपण? Why we discuss? Generally, for in the process of fermentation, it requires at least 5 to 6 hours of times. For the preparation of idli, generally the powder, rice powder, is generally mixed with the water and it kept whole night. But nowadays, instants are available. Take out the powder, mix with the water and prepare immediately. Why this is possible? This is possible only due to the addition of the fast fermentation process. Idli has great demand because easily digestible product, and particularly is the dish of the senior citizens, because senior citizen needs a product which can be easily digestible. Dosa, uttapam, jalebi, angi, some products are of the other Asian countries. It is a breakfast in Asian countries like India, Nepal, Pakistan, Tibet, etc. These are the product which is mostly used, where the fermentation technology is used. No. For the preparation of idli, the fungus which is important, which is discovered, is Turulopsis candida. We add the powder of Turulopsis candida in the powder, in the mixture, then fermentation becomes faster. Not necessary to keep a longer time for fermentation. Rhizosphorum pullulans. For preparation of jalebi, आपली जलेबीला काय करतो आधी? तो रात्री भिजवून ठेवतो, मग सकाळी तुम्हाला तो गरम गरम सांगतो, काढून देतो. त्या रात्रभरात काय होते माहिती आहे का आपल्याला? कधी ते प्लेट उघडी आहे, काय आहे, तो फेकून थोडी देणार. पण त्याला म्हटलं की आताच पावडर भिजव आणि त्याच्यात सॅकरोमायसिस बनियानस टाक, which is available in the market. आणि within hours of time सुरू कर. तो पहा किती चांगली जलेबी देईल तुम्हाला. Fresh products. These are the some of the fungi. In the preparation of jalebi, the Saccharomyces bunians is discovered. Punjabi varieties candida. One of the important is the Saccharomyces cerevisiae. आणि नाशिक मध्ये तुमच्याच आता आजूबाजूला हे सगळं खूप ग्रेप्स वगैरे होतात. And these most of the grapes are used for the process of fermentation in the preparation of different types of wines, where this fungus play important role. Which it allows fast fermentation. I will not take this much more one. These are the products of fermentation. Why these products? What is the importance of this product? Temp is one of the products prepared by Hasseltine in 1965. This product is prepared from inoculated soybeans. It increases the concentration of riboflavin. More riboflavin, more nicotinic acid, more vitamin B12, and that improves the texture of the food and help in easy digestion. This is also used here. Also, these fungi involved in the humus formation. This is the important part. कुळा कचरा हे सगळं आपल्याकडे पडलेलं आहे. आपण काय करतो? तुम्ही मी त्याच्या टीनवाला आहे. आपल्या घराच्या बाहेर कचरा टाकतो आणि आपल्याला वाटतं तो कचरा कोणतरी न्यायला पाहिजे इथून, आणि तो कुठून नेऊन टाकायला पाहिजे? तो कुठून तरी टाकतो. तो कुठून तरी टाकला का तिथून तो कुठून तरी, अरे पण त्याचं डिकम्पोजिशन व्हायला पाहिजे. आपल्याला वाटतं आपल्या घराच्या बाहेर गेला ना तिकडे त्याचं काय होतं तो पाच बस. दिस इज द फ्लोरा. सॉइल मायक्रोफ्लोरा, नायट्रोजन फिक्सिंग ब्लू ग्रीन algae, lichen, fungi, rhizopus, aspergillus, penicillium, yeast, mushrooms. Even the flora and fauna, small insect also help in the degradation process. Amoeba, nematids, earthworm, snail. Earthworm is a eco-friend of farmer. Snail, slug, ant, mites, termites. This is the quality of Can you hear me?
Yes, sir.
कुठेतरी कोणी स्लाइड चेंज केली का माझी? ठीक आहे. I will take a 30 second time.
इथून नाही ऑपरेट होणार सर, तुमच्याकडनं होईल.
नाही, तुमच्याकडून ऑपरेट झाली आहे ते कुठून तरी. ठीक आहे. Now this is the fungi play important role in also making the soil good. That is the fertile. It converts the sterile soil into the fertile soil. It improves the quality. This is the humus formation. It change the quality of the soil, because the best soil means soil containing so many nutrients is needed by the plant for the better growth. Thanks.
Thank you, sir.
Thanks for inviting me on this occasion.
Thank you, sir, for your
Also
valuable
express
guidance.
thanks to the MSG College, Malegaon, principal of the college, head of the department, Jadhav sir, invitees, my friend Yogesh Shastri, all other members and coordinator of the NAC, for whom my thanks are due. Thank you.
Thank you, sir. Thank you for your valuable guidance. You have explained all the aspects of fungi in very simple way, which help us to understand the fungi. Thank you very much. Now, I would like to invite Dr. Praveen Nikam for the vote of thanks. Praveen.
Good afternoon, all of you. Respected principal, all the dignitaries and participants in this virtual national webinar. It is an honor for me to have opportunity to give a vote of thanks on this occasion. On the behalf of the Department of Botany and IQAC, I express my sincere gratitude to respected principal Dr. Dept Shirude sir for giving us the opportunity to organize this event. I also sincerely thanks to our vice principal Dr. Nikam sir and academic coordinator Dr. N.B. Bachhav sir for providing support to make this national webinar successful. I would also like to express my sincere thanks to today's resource persons, who both are eminent personalities, Dr. Adarsh Kumar and Dr. M.N. Bhajbhuje sir, for giving excellent, informative and fruitful lecture on Archegoniate and beneficial aspects of fungi respectively. I extend my sincere gratitude to ISC coordinator, Professor Sai sir, NAC coordinator, Dr. Mukund Bhandari sir, Registrar I.H. Shelar sir, H. HOD of Department of Botany, Professor Wadi Sorone sir, uh, Head of the Department, Dr. J.T. Yadav sir, for their help and support to organize this national webinar.
Sonu,
I must not
yeah.
forget to thank the organizing committee for working hard from last few days
देखो
to make this national webinar
अपन
successful
ये भी
and
ये
fruitful.
सेमिनार कर रहे थे ना,
I congratulate
ये देखो
all
फोटो
the participants
ये
for
ऐसे
their active participation.
तो जब अपन नेशनल ये वगैरह
Thank
लिखते हैं
you
ना,
once
नेशनल
again.
चालू था तो थोड़ा सा कम हो जाता है कट से।
Thank you, one and all.
तो तुम लोग क्या लगता है बिगड़ गए ऐसे नहीं।
With the with the permission of chair, the webinar is end now.